Okay, welcome to Chapter 13 in the continuing saga. When last we left, we were talking about respiration, and I'm going to direct you to plate 55. Let's start up here at the muscle. This is where metabolism is occurring, and CO2 is being produced. High concentrations within the cell diffuse into the blood. Conversely, high concentrations of oxygen, which are being delivered to this metabolizing tissue, the diffusion gradient is from hemoglobin into the muscle. So these show the diffusional pathways. So there's the hemoglobin molecule, there's oxygen coming off, there's CO2 being produced and diffusing in. And I'll point to this little figure right here, which is a cartoon representation of an enzyme called carbonic anhydrase. So you had to take all of that chemistry right for a reason. And if I can find that reason. Here's our reaction. CO2 is going to combine with water to form a relatively unstable acid. This is carbonic acid. This rapidly dissociates into a hydrogen ion and a bicarbonate ion. If this reaction is catalyzed, that is, if there's an enzyme which facilitates this motion and movement, that will effectively decrease the amount of CO2, increasing the amount of covalently bound CO2. The net effect is to reduce the amount of CO2 or to reduce the dissolved CO2. That acts as a driving force to pull the CO2 out of the muscle. So here you've got an enzyme present in small amounts, but it catalyzes. It makes the reaction move more rapidly. What that is going to do is facilitate unloading of CO2 at the tissues. So there this enzyme is right here, and it's pulling it. Carbonic anhydrase is present in high concentrations inside the red blood cell. So it's pulling the CO2 into the red blood cell. Hemoglobin is a protein, and as such can act as a buffer for the hydrogen ion. So you see this, here's the bicarbonate ion, it's being broken, or here's the carbonic acid compound, which is broken into the bicarbonate. And then see this hydrogen ion is being buffered or picked up by the negative amino groups on hemoglobin. So not only is hemoglobin involved in CO2 transport, carbamino hemoglobin, it's involved in oxygen transport bound to the heme groups. It also serves as an incredibly important buffer. So what happens then at the lungs? Absolutely the reverse. Here's the hemoglobin molecule with the hydrogen ion. You now have an environment which is dedicated to removal of CO2. So CO2 is being pulled off, blown out of the lungs. Carbonic anhydrase acts in the opposite direction. It takes this bicarbonate ion and it takes this hydrogen ion. They combine and form CO2, which in the lung is eliminated relatively easily. So it is this interaction of hemoglobin, CO2, and the presence of a carbonic anhydrase, which facilitates the removal of CO2. You always need to think of CO2 as being an acid. how do we control breathing? Let's go to the next plate, plate 56. And up at the top of the plate, you have ventilation, tidal volume. Recall that's the volume of air that comes in with each breath times the rate to give minute ventilation. And this is an increased ventilatory rate and increased ventilatory volume. So this individual is going through an increase in ventilation. The question is, does the increase in ventilation match the increased need in demand? In a normally functioning, highly regulated individual, as your demand for oxygen increases, and subsequently your demand for removal of CO2 is increased, your ventilation is elevated. Minute ventilation goes up to accommodate that. So what is it that we're sensing? We have both oxygen receptors and CO2 receptors, and if I can get this page to behave. Let's look first at the central receptors. So that is up here in this diagram. Central receptors for blood gases are focused primarily on the regulation of carbon dioxide. And you will find that your ability, much tighter control over CO2 than oxygen. In the normal environment, it's 21% O2. You can drop the percent oxygen from 21% to 15 and have very little effect on the ventilatory response. However, if you were to increase the CO2 by that amount, that has profound effects on ventilation. And in fact, CO2 is a form of euthanasia. Concentrations of CO2 around 30% are producing narcosis, sleep-induced response from which you will not recover unless those CO2 levels are corrected. So there's this tremendous drive. Your whole body is orchestrated to keep CO2 levels in a very tightly confined, narrow window. How do they sense that? One of the sensors is located in the central nervous system in the medulla. It responds to changes in CO2, but very indirectly. We have a relatively impermeable membrane between the central nervous system and the vascular blood supply. That's what's called the blood-brain barrier. And you think about it as a protective mechanism. You want to be very careful about what diffuses into the neural tissue. So under normal circumstances, in normal non-pathological tissue, this blood-brain barrier is very tight. It's impermeable to hydrogen ions. It's impermeable to acids, particularly acids that might be generated due to elevations in CO2 from exercise. What we see then is a membrane that's impermeable to hydrogen ions but very permeable to CO2. CO2 is in equilibrium between the blood and the cerebral spinal fluid. The cerebral spinal fluid has relatively low concentrations of proteins. Proteins are buffers. So with low concentrations of buffers, this CO2 combines with water to form carbonic acid, and then the bicarbonate and the hydrogen ion. Because the protein concentrations are low, that hydrogen ion is not buffered. And it is that hydrogen ion which stimulates nervous cells in the medulla, in the central nervous system. So the immediate response is to hydrogen ion, but this hydrogen ion, where did that come from? It came from that CO2. So we talk about central chemoreceptors responding to CO2. They are in fact responding to an acidosis in the cerebral spinal fluid. Stimulation of those receptors in the medulla produce an increase in ventilation. That is driven by the respiratory center. The respiratory center lies in the medulla. So the medulla, as you will learn in lab, is a distal portion of the brain. In fact, with only the medulla intact, so if you were to chop off, you were to decerebrate the individual, they would still be able to breathe. Now, there is obviously modulation from the central nervous system from higher centers, but really the central control of respiration is driven through the medulla. Stimulation of these central receptors and through the inspiratory center causes an increase in rate and an increase in volume. So that's exactly what you see here is an increase in PCO2. So here's an individual that starts exercising. The CO2 go to the brain. They diffuse into the tissue. They drive the inspiratory center. Ventilation goes up to correct the PCO2, to decrease PCO2. Well, in addition to CO2 and oxygen and CO2 and hydrogen ion receptors, we also have chemoreceptors that sense oxygen. These are chemical receptors. I talked about baroreceptors, baro meaning pressure. That was blood pressure. Now we are talking about a separate set of receptors that sense O2 levels. These are located in the aorta and carotid, like the baroreceptors, but their involvement is sensation of O2. So in this little cartoon, this individual is at high altitude, hiked up to Big Bear, 6,000–7,000 feet. The percent O2 at Big Bear is still 21%, but it's 20% of a fewer number of molecules. So in this circumstance, the amount of oxygen, which is the total amount of oxygen that's available, is reduced. We refer to that as a hypoxia. So this individual has gone into a hypoxic environment, breathing air that has fewer number of oxygen molecules. That produces a decrease in the partial pressure of oxygen, a decrease in PO2. These receptors in the aorta and the carotid perceive a decrease in PO2, send that information to the respiratory center. Same respiratory center that was being driven by hydrogen ions, but a different set of nerves. From the inspiratory center, nerves emanate, just as they did up above, to the lungs to increase respiration. And what you see is an increase in ventilation to elevate the PO2. Now consider what happens in a hypoxic environment. You are at high altitude. You are exposed to a hypoxic situation. Decreased PO2. That stimulates chemoreceptors to increase ventilation. Increased ventilation elevates the PO2, but what's it going to do to PCO2? I said your brain was exquisitely sensitive to changes in PCO2, and I gave the example of elevated levels of carbon dioxide. Because of the tight regulation of CO2, you are also faced with decreases and regulating you want to prohibit too much decrease in PCO2. So it's a quandary. In fact, decreases in PCO2 blunt the ventilatory response that's being driven by hypoxia. So you don't breathe effectively enough to elevate the PO2, but you're guarding because you don't want to decrease the PCO2. This is what we talk about, blowing off CO2. When CO2 falls below normal levels because of an increase in ventilation, we refer to that as a hyperventilation. Distinguish that from exercise. When you're exercising, CO2s are going up, PO2s are going down. You want to correct that, so you increase your ventilatory rate. That brings PO2s back up, brings PCO2s back down. That is, it brings them back to a controlled environment. That increase in ventilation is not hyperventilation. We give it another great name. You'll like this. That's tachypnea. Tachy meaning increase, pnea meaning rate of breathing. So during exercise, we all go through a stage of tachypnea. Our ventilatory rates and depths go up. But because we maintain O2 and CO2 in a normally very regulated, tight region, that is not hyperventilation. So, again, hyperventilation. I hyperventilate to increase my O2, and the net effect is it blows off excess CO2. And that disturbs acid-base balance. So in the same equation, if you hyperventilate and eliminate this, so that goes out the lungs, that pulls the reaction to the left. That's going to decrease hydrogen ions. That is going to make it more alkalotic. And because it's due to an increase in ventilation, we refer to that as a respiratory alkalosis. Some of you can be profoundly impacted by that. You go and you get what's called acute mountain sickness. Raging headaches, nausea, maybe vomiting. It can be incapacitating and in some cases life-threatening. So that's an example of respiratory alkalosis. What we see clinically more often is animals that come in a depressed state. Hit by a car, they're in shock, and or you're going to do surgery on them. And so you artificially induce a depression of the central nervous system. You give them drugs. In those individuals, they are not increasing their ventilation. They are decreasing their ventilation. Because it's not being controlled, the PO2 levels are dropping, so we say these individuals are becoming hypoxemic. The oxygen in the blood is decreasing. Because they're not ventilating adequately, PCO2 goes up. That is a hypercapnia. Just the opposite from hyperventilation. This is hypoventilation. Animals are not adequately ventilating to deal with the O2 or to get rid of the CO2. Elevations in CO2 drive that reaction back to the right, and you become acidotic. So this is an example of respiratory acidosis. And uncorrected, you can die from it. So one of the big challenges in anesthesia is to maintain the ventilation appropriately so that O2 and CO2 levels are adequate. Now, commonly what's done is you feed a tank with pure oxygen. That's 100% O2. You still should be measuring CO2. High-end clinics will do blood gas analyses. It's not free. Very expensive to do blood gas analyses. But it, in fact, is the best way for you to get a handle on whether the animal is hyperventilating or hypoventilating. So blood gas, that's your friend. Can you hold your breath till you die? You know, I kind of threatened that at a younger age. I'm going to hold my breath till I die. Well, yeah, sort of. Not in air, you're not going to be able to do that because of the drive to increase ventilation due to elevations in CO2. So even if you hyperventilate, okay, you blow off the CO2, you elevate the oxygen, and then you hold your breath. Eventually what happens is the CO2 level builds up and forces you to inspire. The problem comes if you're underwater when that happens. So individuals that hyperventilate before taking a dive can run the risk of getting a forced ventilation while they're underwater, and that is not a good thing. Plate 57 kind of talks about hypoxia and high altitude. Give me a second as I come back to the plate 57. Where are you? One of the neat things about hemoglobin is that it changes color as it becomes oxygenated and deoxygenated. Red when it's fully oxygenated, a cyan or purple blue when it's deoxygenated. So let's look at some of these changes. Here's hypoxic hypoxia. A hypoxia, a decrease in oxygen in the lungs due to the fact that you are exposed to a low O2 environment. So this is high altitude. We talked about the stimulation, the increase in ventilation, and the symptoms of altitude sickness. Now what happens is adjustments to that, one of which is a stimulation of a hormone called erythropoietin. So if you go down to this figure right here, you have the kidney, which is normally presented with about 30% of the blood flow at rest. So it's an organ that is poised to have an important role in oxygen sensation. It sees a lot of blood that has oxygen in it or not. Well, there are cells, juxtaglomerular cells, which produce a hormone called erythropoietin. Poietin from the Greek meaning to produce, erythro meaning red cells. This hormone, released, it goes to the bone marrow where it stimulates increased number of red cells. So a process that may take 7 to 10 days. Your response is, I'm in a hypoxic environment, I need more oxygen. One of the ways to do that is more red cells, more hemoglobin. Now let's take a clinical case. Animal comes in, gums are white. You take a blood sample and you see that the animal is anemic. It has low numbers of red cells. What color are the gums going to be? They're going to be a little white, depending on the degree of anemia. Are they going to be deoxygenated? Are they going to be purple and blue? Or are they going to be pink? Good test question. In anemic hypoxia, you're in an environment that has normal O2, but you don't have the number of hemoglobin molecules. And in this cartoon, it's represented by these little sailboats are hemoglobin molecules. So this anemic individual has a reduced concentration of hemoglobin. The net effect is this animal is not getting adequate amounts of O2. The response is an increase in ventilation. So invariably, anemic individuals show a hyperventilation. They are trying to get oxygen levels up normally, but remember that very little O2 is dissolved. The majority of O2 transport is by hemoglobin. You don't have hemoglobin, or you have reduced amounts in an anemic patient. So in fact, all the hemoglobin they have is usually saturated, that is, usually red. But because they have so little of it, they're still oxygen starved. The gums may appear pink or even white, depending on how little red cells they have. And CO2 levels, these animals tend to have lower than normal CO2. So they are, in fact, hyperventilating. Treatment for anemic patients, more red cells, either a transfusion or addition of erythropoietin. In fact, the development of human erythropoietin, recombinant EPO, was driven primarily by the kidney failure business. Human medicine, a common sequelae to kidney failure is an anemia. Why? Kidney's not working, no erythropoietin being produced, no red cells. So the pharmaceutical company came up with this great compound, a protein, a synthetic protein identical to humans, right? Recombinant DNA technology. The advantage is no immune response. You get to take human EPO, and has really been a remarkable boon to kidney patients. And as you might also expect, it's been a problem with abuse. Cyclists, the cycling sports medicine group has been implicated, highly implicated in doping by the use of this recombinant DNA. These individuals present with elevated concentrations of red cells. They take so much erythropoietin that they drive up red cell production to such levels that it affects viscosity. And these individuals can die from heart failure from trying to pump this sludge through. Since I've made a point about the kidney, I'd like to focus our attention now on kidney structure and function. Again, 30% of the cardiac output in a resting individual goes to the kidney. On plate 58, it mentions some numbers in terms of the volumes that are filtered, the volumes of urine that are produced. It's not important that you memorize those volumes, just to have a comprehension of the magnitude, the enormity of what gets processed in the kidneys. What do the kidneys do? They filter. Well, they also produce erythropoietin, but they filter. Oh. You're going to look at the anatomy in the lab, so I'm not going to make a big emphasis on the normal anatomy, but I want you to kind of review that you have a renal artery and vein. So this is where paired organs, blood flow comes into the arteries, leaves by the vein. And in the kidney is divided up into a capsular or outer portion that is a tough overlying tissue. Under that is the cortex. And then the central layer is called the medulla. In this diagram, you see that there's blood coming in, 1,300, 1,299 coming out. That is, the amount of blood that's coming back is very close to the amount that goes in. So there's a process occurring. You're filtering it, but you're also reabsorbing most of that. And that process occurs in the functional unit of the kidney called the nephron. The nephron consists of Bowman's capsule, and I think there's this diagram up here shows the orientation of these different units within the cortex and medulla, and you need an appreciation for that. But let's go look at the microscopic level. You'll do this in lab. Incredibly convex or complicated, convoluted set of tubules, particularly in the outer cortex. If we look and dissect this a little more carefully, you get an image that looks like this. A renal nephron that is composed of Bowman's capsule, a proximal tubule, a distal tubule, collecting ducts, and then a very pronounced loop. All right, so up here is the proximal. Bowman's capsule is right about there. So that's that figure right there. Those are the proximal tubules. You'll see then this drop of this nephron tubule down into the medulla, back up into another series of convoluted tubes. So there are proximal and distal convoluted tubes. The distal convoluted tubules dump into the collecting ducts, which pass again through the medulla and exit out through the ureter. Material from the blood is filtered into the nephron. It circulates through the proximal tubule, down this loop, back up this loop, through the distal tubule, and then through the collecting ducts. There is a blood supply into Bowman's capsule and a blood supply out from the capsule, both which are arteriolar in nature. So here's an example in which you have an artery and a capillary bed and another artery. Very, very unusual. Smooth muscle, lots of smooth muscle under sympathetic control. Blood coming in this arteriole, so it's coming in, so we're going to call this an afferent arteriole. And blood comes out of Bowman's capsule in an efferent, so think of E as being exit. So efferent arterioles take blood away from the capsule, afferent bring it in. This combination of Bowman's capsule, so there's your anatomist, and the blood supply is called a glomerulus. So when we talk about looking in lab, you're going to look at the glomeruli of nephrons and you're going to see this kind of bulbous structure. It consists both of cells from Bowman's capsule, which is part of the nephron, and cells of the afferent, efferent arterioles and the glomerular capillaries. And we're going to look at detail in what's happening in the glomerulus. The last thing I need to talk about, let's go back to this figure right here. This loop of nephron that drops down into the medulla, named after anatomists, this is called the loop of Henle and becomes incredibly important in the ability to produce concentrated urine. So the loop of Henle is part of the nephron. We also have capillaries that are not only associated with the glomerulus, but we also have capillary beds that are next to these tubules. These are peritubules. peritubular capillaries. These capillaries are glomerular capillaries. The peritubular capillaries arise off of the efferent arteriole. So there's another capillary bed here, which is called these peritubular capillaries. And then we have a unique set of peritubular capillaries which follow the loop of Henle, and they are given the name vasa recta. So the vasa recta are modified peritubular capillaries that follow the nephron's loop of Henle. And we'll come back and talk about those in more detail. But let's take a break.